Thank you very much. Thank you very much, Ichiro, for the kind introduction, and also thank you very much for organizing this cozy workshop. So I'm very pleased to present our effort over the past 5 years to dissect the role of non-coding DNA. So as you know, our genome consists of 3 billion letters of A, G, T, and C. This encodes not only the difference between male and female or the appearance, but also some lifestyles and also susceptibility to diseases. And also it is said that our knowledge, current knowledge, is the tip of an iceberg, and actual human genome variation is more complex than we thought 20 years ago we opened up our genome. So the outline of today's talk is, first of all, I would like to explain what is non-coding DNA, if you are not familiar with. Then I would like to explain how to link with diseases, and here we would use some epidemiological or population genetics approach called genome-wide association study and quantitative trait locus mapping. And then I would demonstrate how to interpret the function of those discovered non-coding DNAs with 3 examples. And here, those three genetic loci are discovered through different genome association studies. So to study the non-coding DNA, we first need to understand what actually DNA, the mechanism of DNA transcription and translation. So our DNA has a helical structure consisting of 2 linear sequences of A, T, G, and C, and here the RNA polymerase multi-protein complex that unwind DNA and synthesize premature messenger RNA using the templated strand of coding sequence, which is actually a copy of the DNA sequence. And then this pre-mRNA is spliced and processed into mRNA, then translocated from cell nucleus to cell cytoplasm and translated into amino acid chain and finally into a protein. This process is so-called central dogma of biology. So here, some mutations may alter this process to create some genetic diversity between us. And some mutations do nothing because the base substitution at messenger RNA level doesn't change the amino acids, so there's no protein difference. But some mutations do change the amino acid or even terminate the translation, so which results in inappropriate protein or premature protein or not even no protein being made. So the stretch of DNA that can code for DNA is called gene. And you might think genes are sequentially existing in the genome, but in reality, genes are scattered over our genome. Between 2 genes, there is a called intergenic region. Besides, genes are usually composed of multiple exons and introns, and because only exons can code for a protein, so overall, the coding DNA accounts for only 3% of our genome, and rest are non-coding DNA. In addition, our genes are not expressed at the same time, so around 98 or 99% of our genome actually doesn't code for protein at a certain point. To So previously, researchers thought those non-coding DNAs are redundant and therefore called junk DNA, but it wasn't. So to find the function of non-coding DNA, we particularly focus on genetic variation between us. So the most common form of genetic variation is called single nucleotide polymorphism, or SNP. So SNP is basically a base substitution at exactly the same location of our genome between two or many people. And the distribution of the distance between two SNPs in the worldwide population with a certain frequency shows that the SNP exists once in 150 base pair on average. And as we expect that those SNPs within the coding sequence only accounts for 1 to 2% of the genome, and others are all non-coding SNPs. To use those SNPs, in 2002, a Japanese research group invented a very strong epidemiological approach called Genome Association Study, or GWAS, to determine, to map genetic determinants of diseases on our genome. So in GWAS, we collect many samples, both from healthy controls and disease cases. For example, myocardial infarction studied in 2002. And then we look at the risk allele frequency difference at the SNP locus. And in this example, G allele is significantly overrepresented in case population. So this allele in the SNP can be a risk for a disease. In GWAS, we scan all SNPs in the genome from chromosome 1 through chromosome 22, and sometimes X chromosome. And the figure shows, the point in the figure shows each SNP, and the y-axis shows statistical significance. So the higher, the more likely to be disease-associated SNP. And this plot is often called Manhattan plot, so you must be familiar with. So if you find the Empire State Building or One World Trade Center on this plot, this study has some positive findings. And the European Bioinformatics Institute has been collecting those genetic associations shown in the diagram as a GWAS catalog. And the results is growing and growing. And in 2024, more than 400,000 associations in 30,000 common complex traits were reported in the catalog. And it is not surprisingly, many of them, actually more than 90% of them were discovered in non-coding sequence rather than coding sequence, which alters protein structure. So the simple question here is, what are they doing? So let's jump into the first example, which is an Alzheimer's disease genome-wide association study conducted in 2021, and which I was involved. Although the highest association was discovered at the ApoE gene, driven by 2 missense mutations. The second highest association is discovered around BIN1 gene. And if we look at the location closely, the highest association is attained by this SNP, and this underneath there's no coding sequence. The SNP is upstream of BIN1 gene, and there's and this SNP is non-coding SNP. To understand the mechanism of why we find those non-coding SNPs, we need to understand we need to know the existence of chromatin, which is a DNA protein complex. And you might be familiar with the picture of chromosome like this, but in reality, this image only happens when cells are dividing. And in most of the time, chromosomes are existing like noodles in a bowl. Here, a bowl is a cell nucleus. And DNA is normally packed and wrapped around the protein called histone, the cylinder-shaped protein, and to form a nucleosome. And you can see the nucleosome by eyes if you have a fancy microscope. And so DNA is usually tightly packed and which form closed chromatin. But in some region, DNA is loosely wrapped around histone, or even DNA is naked. This region is called open chromatin region or chromatin accessibility region, where naked DNA is bound by a very important protein called transcription factors, with some preferable sequencing motifs. So those transcription factors are clustered in a region called enhancer, which enhance the gene expression. And those enhancers are scattered around the genome in the linear genome. But in reality, those the DNA bends back on itself, and those transcription factors are clustered and cooperatively initiate gene expression. So you can now imagine that if there is a SNP in the enhancer region, those SNPs tend to disrupt the transcription factor binding motifs, and then which often reduce the transcription, or sometimes the SNP mutation stops the transcription. To map those SNPs that alter the gene regulation, we again collect many samples from the people, and then correlate it. We check the correlation between underlying genotype and gene expression, or underlying genotype with chromatin accessibility, which captures the enhancer activity. And if we find some correlation between them, then we call them expression QTL, or in short EQTL, or chromatin accessibility QTL, or CAQTL in short. And here genotype indicates a pair of two alleles at the SNP location, one from your father and one from your mother. So there are three different genotype groups in the population. So we can split people in this room into three different groups at some SNP here. So in parallel to the Alzheimer's disease genome-wide associations, we studied EQTLs of microglia, human primary microglia. So a microglia is a brain resident macrophage and accounts for 10 to 15% of brain cells, and is implicated in neurodegenerative diseases such as Alzheimer's or Parkinson's diseases. Our collaborators in neuroscience department in the University of Cambridge collected those microglia from 141 neurosurgery patients. I think it was really painful procedure, I think. So then here is again the association of Alzheimer's disease. The second panel shows the BIN1 EQTL signal observed from the data set. So the pattern of the association is really similar, meaning BIN1 EQTL is involved in Alzheimer's disease onset. And we've also measured chromatin accessibilities from 5 neurosurgery patients, and we confirmed that this top hit variant is located in the chromatin accessibility regions. Therefore, it alters some gene regulation through disrupting some transcription factor motif. And also we've differentiated 89 macrophages from iPS cells, and we look at the genetic variation of chromatin accessibility between 3 different genotype groups at this top hit variant. And here the diagram shows chromatin accessibility segregated and averaged by the 3 different genotype groups here. And the top associated variant for Alzheimer's disease is sitting at the shoulder of this peak. So by the way, the peak is higher means the chromatin accessibility is also higher in this case. So T allele increased the chromatin accessibility. Actually, there is a MEF2A transcription factor binding motif underlying this SNP, and there, and the T allele is more preferable than C in this example. And we've also scanned multiple transcription factor binding motifs around this peak, and we've also found that SPY1, which is a key lineage transcription factor for macrophages and microglia. So what we learned from the data set is if you have T allele, MEF2A binding affinity is going up, and possibly SP1I as well. And then BIM1 expression is increased. And finally, Alzheimer's disease risk is going up. So then let's move on to the next example, which is a BLK locus discovered through systemic lupus erythematosus genome-wide association studies. So this locus is also known to be associated with other autoimmune diseases such as rheumatoid arthritis or Kawasaki disease and so on. There are multiple non-coding variants associated with SLE, and those are located in between 2 genes, FAM167A and BLK. To understand the mechanism underlying this locus, we further need to know the multiple layers of 3D genome organization. So as I told that our genome is existing like noodles in cell nucleus, and which forms both non-active and active compartments within the cell nucleus. And those compartments are partly fixed by important factor CTCF or CCCTC binding factors, and as an anchor of DNA loops. Within the loop, as I told, that transcription factors can interact with transcription start site to initiate and promote gene expression. To study to capture the 3D genome organization, previously we developed a Bayesian method called pairwise hierarchical model. I'm not going to the detail of the method today, but the rationale here is there are 2 chromatin accessibility peaks, and both the accessibility is high for some individual and low for the other individual, and this is consistent across many people. Then we believe that there are some interaction between those 2 chromatin accessibility regions. And using this information, we were able to pinpoint a variant located in between those 2 regions. although previously 3 different SNPs around the transcription start site of BLK were implicated to SLE and rheumatoid arthritis. And this SNP is found in chromatin accessibility peak, in the middle of chromatin accessibility peak, and we've discovered that this peak is actually positively interacting with other regions at BLK side and negatively interacted with the FAM167A side. So for the validation of this locus, and we found that this SNP is actually, the variant is actually not SNP, but one base insertion variant, and the extra G in the alternative sequence significantly reduced the CTCF binding motif. And we've also confirmed that with the external data set that CTCF binding is significantly reduced by this extra G. So our collaborator Andy Knights created 2 deletion lines using the CRISPR gene engineering technique, and then to confirm that the accessibility at the target peak as well as the BLK expressions are both downregulated by this deletion. So the hypothetical model here is due to the one base pair insertion, CTCF binding site is disrupted, then CTCF is detached, then the loop structure is changed, and the TFs that were interacting with BLK is now interacting with FAM167A to create a bidirectional expression pattern in this region. And somehow this is associated with multiple autoimmune diseases. So the last example is GWAS for COVID-19, which discovered OS1 locus in the chromosome 12. To understand the mechanism underlying this locus, we should know that the use of enhancers is actually cell type specific. So in the bulk population, bulk cells, there are multiple enhancers discovered through the chromatin accessibility quantification, but in reality, some enhancer is only active in one cell type, but not the others. So to study the cell type specific dynamics of gene regulation under the infection, we developed some experimental system using a human skin fibroblast from 6 to 8 donors, and then we stimulated cells with interferon beta or poly-IC. Those stimuli are known to be mimic cells being infected by a virus. And then to study the cell type dynamics, we sequenced expression level of each cell at a time. And here is the result of cells under virus infection. And each dot shows the cell of more than 20,000 cells. And you can see the trajectories of cell stimulation, one for interferon beta and the other for poly-IC, and some similarity between those two after several hours of experiment. And to use this information, we discovered that the COVID-19 GWAS association in the OS1 locus is actually an EQTL under the virus infection. So OS1 is highly expressed in the cells with certain time point and after cell stimulation, which means cells are reacting for some viruses. And the expression difference between the A allele and G allele, both in this the target SNP, showed that if you have A allele, OS1 expression and the virus infection goes down, and the risk of COVID-19 is increasing. Because OS1 is involved in the cascade of virus RNA degradation and clearance in the cell. So cells with less OS1 protein, because of less OS1 expression, results in the cells are not able to fight with coronavirus, for example, and then you get some higher risk of COVID-19. So in summary, while coding DNA alters and creates the different types of genes, non-coding DNA tends to alter quantity of gene or transcripts. And here I introduced three ways that non-coding DNA alters quantity of transcription, and which all modulating disease risks. And the endeavor to explore the non-coding DNA has just begun, and we would continue to work on it. So finally, I would like to thank people at the Sanger Institute and also University of Cambridge, and also donors who provided precious samples. Thank you very much, and I'm happy to take your comments and questions and comments. Thank you.
Thank you.
I have to ask the first question always. So the not being in the GWAS field, the associated loci or SNPs, how do they interact? Are there interactions that you see? Like some of those peaks, are those cooperating in regulating the expression of different genes in a deleterious fashion?
You mean one SNP in the coding sequence and the other in non-coding sequence, or interaction between two SNPs, you mean?
Yeah, no, interaction between SNPs that are completely on different chromosomes, but are both highly significant.
Yeah.
I think. Do you see them associated with each other sometimes, or are there patterns?
Between SNPs, I don't think so, because they are on the different chromosomes. So in principle, they should be orthogonal. Or
yeah, it wouldn't be a haplotype, but it would be a functional association.
So to some trait.
Yeah,
I think there are many SNPs that acts in cis or trans. So trans SNPs alters gene expression on different chromosomes via cis regulation of some genes on the same chromosome. So for example, transcription factor gene has an EQTL, then this SNP alters the expression level of transcription factor, then this affects all other genes downstream of the transcription factor.
I'm referring to where you showed the Manhattan plots, and there was one locus that was the highest, but you then looked at the second highest.
Oh, okay.
So I'm just curious, how does that ever occur? And this is again,
so you mean
yeah.
here?
So ApoE and BIN1, do they cooperate in a way? Like, do
No,
they co-occur?
they are completely independent.
So
So-
if you have the so-called bad variant at both loci,
Mhm.
do you get a even higher risk, or?
I think if you assume that the risk is additive, then you can add. If you have some risk allele for B1 and risk allele for ApoE, your risk is just double. But it depends on, I think, the model you believe. But in general, it should be additive, I think.
Thank you.
So thank you very much. So I have one question about this data. So you focus on macrophage.
Yeah.
Why you are interested in macrophage? Because Alzheimer is a neurodegenerative disease. So
Yes. So
before taking this approach, you may check the expression of the gene of each cell types, or how you
there
can-
is some studies that check the enrichment of expression for various diseases, and I think macrophage is one of the most enriched cell type for the Alzheimer's disease. If you use, for example, LDSC or LD score regression or something.
And so you GWAS data says several other SNPs associated with these diseases. So all SNPs can explain this macrophage gene expression or different cell
No,
types.
unfortunately, no. So there are some SNPs can be explained by macrophages, but not all. So it is more complicated than we thought.
I see. Thank you.
Any questions? So this mechanism is one of possible many adjustment to cause Alzheimer's disease. And this is one example of adjustment or making one single error in the gene causing Alzheimer's.
So usually we discovered many SNPs through genome-wide association studies, but all of them have very small risks. So, for example, this BIN1 SNP has only 1 point times higher risk than the normal population.
So
even if
double,
you have,
1.1 meaning
sorry, 1.2, sorry.
1.2, 20% more
Yeah,
likely
just
to develop
20%.
Alzheimer's disease. But 20%, that's quite a lot. If you have 5, and then that's double.
Yeah, but unfortunately, we don't see much of those very strong effect size. And usually, top hit is like three times higher risk, then 1.2, then rests 1.1, then 1.05, and 1.01 or something. So
you do not have to be scary about this as much. Maybe just
But
one single SNP.
the scary thing is we haven't discovered all
The others.
all other loci.
I have one question for your COVID project, and actually I have one COVID works done by myself, and I have one question. Why did you choose fibroblasts for use for analyzing the GBUS? Because COVID virus usually combines to ACE receptors, and the ACE receptors usually expressed in the endothelial cells. So I want to know some rationale why you focus on the fibroblasts.
So it is a very good question, I think, and this project has been started before COVID.
Oh, really?
So we don't expect anything about COVID at that time. We didn't. And but we thought the reason why we chose fibroblast is that the innate immune response with those stimuli should be similar to
Immune
many
cells?
other cell
Oh.
types, and the fibroblast was the easiest way to get some materials at Sanger Institute at that point. So actually, we haven't designed this system for
Specific
COVID.
for-
No, no.
Okay, thank you so much.
Anyone else? So again, from a person, amateur person without knowledge of gene, I'm just curious. There was a discussion about cis effect, trans effect. This little tiny 1-point mutation, meaning error, can affect all the way to the other end, and it make an adjustment. Like a tiny little earthquake in Japan, and it really changes the position of Manhattan kind of thing. Very far away, because far away, chromosome, different building, just one accident in one floor does affect that Empire State Building, 50th floor. So how can you find out the connection between those very far away? So I'm just curious. It seems to be very difficult to find an association.
I don't think we in the Manhattan plot. This is just an association between one SNP and some disease, for example, disease occurrence. So we don't know the underlying cascade, just an association between one SNP in some chromosome and to some disease. We are looking at.
The computer does the job. Or
yeah,
you don't write the mathematics,
we don't.
but the
We don't
computer
calculate by
put
mathematics.
all the information, macro information, what is the mega information into the computer, and then try to find out your future spouse. Probably it's an OMI system, probably, and so that's what they do. Computer tells you, it's your future wife, blah blah blah, by putting all the information.
I don't think we can do this with genome-wide associations, but
But the association was found by putting
yeah.
in huge data number, and
Yeah,
the computer
huge
does tell you,
sample
oh,
size.
this one really making this happen.
yes.
But that's amazing. And then you actually prove that by cutting the portion out, and the effect disappears, and those kind of stuff.
So we can use CRISPR engineering technique to validate, I think.
CRISPR, Cas something.
Cas9,
So,
yes.
yeah, that's a gene technology, which we talked about actually last year. I learned that last year in this seminar. Well, everything is related to each other. Any questions? Okay, so let's move on to the next thing. Thank you very much, Natsuhiko. So Professor Kumasaka.